The overall goal of this procedure is to isolate, grow, and analyze HPV antigen-specific T-cell clones from patients. First, establish a CD8 T-cell line, perform an Enzyme-linked assay, and estimate the frequency of antigen-specific T-cells. After isolating the antigen-specific T-cell clones on the basis of interferon-γ secretion, perform a high-throughput screening interferon-γ ELISpot assay to identify epitope-specific T-cell clones. Continue to characterize the T-cell clones for the minimal, optimal peptide epitope sequences. Also, determine whether the epitope is endogenously processed, identify the HLA restriction element, and assess cross-reactivities to other high-risk HPV types. Ultimately, the goal is to describe novel and clinically relevant HPV T-cell epitopes through the screening and characterization of antigen-specific T-cells from patients.
The main advantage of this technique over existing methods, such as using Coleman-List assay to identify antigen-specific T-cell clone, is that the likelihood of success is much higher.
This method can help answer key questions in the immunology field, such as which portions of HPV do T-cells recognize?
The implications of this technique extend toward therapeutic diagnosis of cervical cancer, because vaccines and immunotherapies can be developed based on the knowledge about T-cell specificities.
This method can provide insight into CD8 T-cell epitopes, and it can also be applied to other systems, such as CD4 T-cell epitopes.